All right, thank you. And thank you to ARM for having us here and giving us the opportunity to present our company. Just some forward-looking statements here, our obligatory slide. So our mission at Teknova is to accelerate the introduction of novel therapeutics, vaccines, and diagnostics, and we do this by providing critical reagents from discovery through process development, through clinical trials, and commercialization. And the three kind of main products that we offer are agar plates, which I know many of you probably look at and think are petri dishes, but to us that live in this world, these are very complex formulations that span from very high throughput screening plates to different antibiotic mixtures that we produce every day. The second line of products is cell culture media. We are one of the leading providers in bacterial cell culture media, providing both research use and clinical use products there. And the last is molecular biology reagents, and this comes from everything from 2 ml tubes of DNA resuspension buffer to complex chromatography solutions that get delivered all the way into 200-liter single-use bags. So the company was actually founded in 1996 by a scientist out of Genentech. His name is Ted Davis. He was there ordering custom reagents from their in-house production, became very frustrated with the fact that he couldn't get the quality and the speed he needed, and he started the company out of his garage. And from that day, for the next 23 years, didn't raise a single amount of capital, no debt, and basically bootstrapped the entire company all the way through that period. He built a phenomenal foundation for us to build on, including 3,000 active customers. So anytime you're any major biotech, pharma, academic, life science tools, or diagnostic company, their stockroom is full of our Teknova reagents. In addition, during that time, he also focused a lot on custom manufacturing. So we do a lot of OEM manufacturing, but we also do custom manufacturing. Customers come to us and say, here's my formulation. I would like it in this container with this QC, and we can get that in production within a couple weeks. That is the foundational difference of our business today and what we continue to build on and scale with over time. So when it comes to cell and gene therapy, there are three major capabilities that Teknova offers that enables us to serve these customers. The first is just what I mentioned, the modular manufacturing platform. The idea that someone could come to us with a detailed formulation. Within a couple weeks, we can procure all the raw materials. We use our 20-plus years of know-how to mix that material, get it into formulation, get it through sterility, and aseptically dispense it and QC it and get it to the customer in weeks instead of months. The second piece is around the scale we offer. So unlike monoclonal antibody therapeutic production, we are operating in the 2,000 liters or less range. So we can do custom reagents in 20 liters all the way up to 2,000 liters and get those to customers very quickly. And the last piece is really this discovery all the way through commercial offering. So when we talk about this, we're really working with our customers very closely as they go through process development and then help them scale through GMP. This is not one of the scenarios where you go work with a group, you find out that they're doing RUO and you want to move to GMP and now you're moved to a different location or different equipment. We scale very seamlessly with the customer. So today I'm going to touch on two major initiatives that we've been putting in place over the last year or so that are now coming to fruition and that we're very excited to talk about today and going forward into 2023. The first is the build out of our manufacturing facility, and that will bring significant capacity to us and allow us to continue to offer products within weeks to customers going forward. And the second is our R&D pipeline, which we've established over the last year, and now we'll be talking about a couple of early access programs today. So from the manufacturing perspective, all of our new facility that's coming online, and probably facility is not the right word to use, this is really a modular manufacturing platform where we focused everything from the design of the clean room to the utilities, to the building, to the quality system, the ERP, the equipment, which we custom designed and built ourselves to be for maximum work order capacity on a daily basis. And so what you see here on the left-hand side of your screen is actually a snapshot of the facility today. You see what we call process trains in there on the left, and then our own custom-made bottle filler on the right. And just give you an idea how this works, we actually, on the other side of that wall, have a matching process train. So we put the powder in the process train on the other side of the wall. That gets formulated and mixed into solution based on our know-how and knowledge of how to put that into solution. Then it gets pumped through the wall and back into what we call the dispensing process train on the right. And that is connected to one of our many dispensing technologies. What you see here is the bottle filler, but obviously we have a tube filler and a bag filler, among other things. While that's dispensing, the formulation train is actually doing self-cleaning and preparing for the next formulation. When the dispensing train is done, then it does its formulation. So you can see we have continuous process of liquid going through the system at all times, allowing us to get the maximum number of work orders done every day. So we're very excited about this facility. We're still on track for our commercial launch of this for research use only by the end of this calendar year, and then clinical certification by mid next year. And this will bring ample capacity to our business and help us scale with the rest of the organizations working in the 2,000 clinical trials happening today. Shifting gears now to the R&D team. So we established an R&D organization about a year ago. Super excited about what we've been able to accomplish in this amount of time. We've talked to our 80-plus cell and gene therapy customers and realized that, while the custom solutions we can provide to them very quickly are enabling, there are significant challenges in bioproduction today, in particular around AAV production. And so we created this group and we said, let's just try and make AAV internally, and we'll get a better feel for how our customers are struggling with this production. And we did that, and they are struggling for the right reasons. This is a very complicated process with a lot of inefficiency and reproducibility issues. Through this process, we identified a couple major bottlenecks that we feel like we can address. And so the first is around what we're calling a buffer screening kit. So at the end of the process, you're trying to separate what we call empty capsids from full capsids, and actually separating full capsids from partial capsids. So you have your virus inside your transgene, and this particular polishing step is pretty complicated, and it varies based on your serotype, your anti-exchange platform, as well as the transgene itself. So every single one of these therapies actually needs a slightly different formulation. We spent the last 6 to 9 months doing a pretty comprehensive DOE to find the boundary zones of all of the different parameters, as you can see on the left-hand side, for the different buffers that could be combined to make the best purification reagent for this process. And through that effort, we've identified a series of buffers that we can now then provide to our gene therapy developers. They can run those buffers with their serotype on their anti-exchange platform with their transgene, and from that data, we can work with them, pull it out, and identify what is the absolute correct custom buffer for them to separate their full and empty capsids and get the highest yield and highest purity possible. This significantly streamlines the process for them as they go through getting this set up in process development. And of course, then as our business continues, they can scale with us over time. We are excited to offer a technology access program or early access program for these products. We have a number of parties that are already interested. We'd like to find more. We're ready to go, so please, if you're interested, come talk to myself or someone on the Teknova team, and we'd be happy to assist you. So, in addition to this, I'm actually going to pop back a slide. If you look on the bottom right-hand corner, you see some green cells, and many of those of you familiar in this space know that is actually GFP, or green fluorescent protein, lighting up those cells. And what we did here is, of course, we used GFP as our transgene in the optimization of these reagents. But I just told you in this section that the transgene, the size of that gene, the charge of that gene, how that gene folds actually changes your reagents. And so one of the challenges in this process development is to identify a way in which you can optimize your reagents with your actual transgene, as GFP is often used as a surrogate in this place. And so we said, what if we, instead of using GFP, we allow customers to use their own transgene and develop a technology that allows us to sense whether or not the gene of interest has been transcribed. And that's exactly what we've been developing. So what you see here is a workflow for how we make that happen. First, we insert the genetic circuitry into the cells, inoculate the cells with the machinery necessary to make this happen. The second day, you apply your virus on top. And the third day, if that virus has the gene of interest in it and it is transcribed, those cells will turn a color. And that allows us to actually optimize the reagents for the gene developer's actual AAV. So in this case, it's great for the reagents. I just said the polishing, but you can imagine across the entire workflow, optimizing your upstream and downstream steps using this functional assay. And the good news is that we've actually made quite a bit of progress on this. So what you're seeing on this slide is actually multiple different AAVs working with our new reporter assay. So on the top, what you see is GFP being expressed as the transgene. So you're seeing the cells as being used with GFP as a transgene. On the bottom, you're seeing a reporter assay. And so you can see that you're actually getting very good correlation between those two different readouts and allowing customers to use their transgene. We are still developing this technology, and again, now we're looking for partners. We have a number of interested parties at the moment, but we're seeking more, and if you're interested, please follow up with me afterwards. So one last piece, over the last 6 to 9 months, we have heard time and again about the limitations of getting Wi-Fi quality water, whether it's for cleaning, whether it's because someone's setting up a new laboratory, or they just need it for rehydration of materials. We are currently building inventory of Wi-Fi quality water that's ISO 13485 compliant that will be in stock in November in 20 and 200 liter bags that will be shipped in a single day. Thank you. And so finally, look, just a quick summary here. Look, Teknova is a leading provider of critical reagents across discovery through clinical and commercial. We offer everything from a vast catalog of products for stock, for discovery work, but really are enabling a lot of different providers in the cell and gene therapy space around custom manufacturing during process development. In addition, we've spun up an R&D group that is obviously doing pretty amazing things. We're very excited about the type of work that's coming out of this, and we really think we can help get these drugs into the clinic faster. And lastly, of course, we do have the water coming in November. So thank you very much.